Good morning, everyone. Welcome. Thank you so much for joining us today for our next edition of our MyDog webinar series. And today we're focusing on parasites, those cryptic critters. I'm so happy to see so many familiar faces. Welcome. Thank you for joining us. To streamline today's presentation, we will go over all your questions at the end of the talk. There is a Q&A up there. Please put all your questions in there and we'll make sure to answer all of them at the end of this talk. I'm very excited to introduce today's speaker, our very own Dr. Mark Yacoub. He's a scientist here with us at MyDog Animal Diagnostics, and he focuses on analyzing shotgun metagenomics data, and he leads our efforts to assemble and investigates the genome of animal parasites and pathogens. Dr. Yacoub earned his PhD in microbiology from the University of California, Riverside in 2024, and his bachelor in microbiology from the University of California, Davis in 2016. His research has primarily centered around the genomics of animal pathogenic bacteria and fungi, as well as those parasites. He's passionate about applying microbial genomes to better understand and treat pathogens posed to animals. And with that, Mark, please take it away.
Thank you, Yanina. Can everyone see my screen here? Is it showing up?
Yes.
Wonderful. Good afternoon. My name is Dr. Mark Yacoub, and today I'll be talking about cryptic critters, which are these parasites, and how at MyDog we use genomics to reveal some common veterinary parasites, but also maybe some lesser known ones that don't show up quite often. So just to get started on some clinical background of why we care about parasites here at MyDog. Parasites affect a wide range of domestic and wild animals, and oftentimes they can cause some important clinical diseases. This can be intestinal disorders. In severe cases, some neurological disorders can be caused by parasites. And importantly, recent studies have shown that 21% of dogs and cats in the USA are infected with intestinal parasites with recurrence for several species. Now, this can actually pose some serious problems to animals, because once an animal has cleared that disease, it will simply recur and come back, which can be very frustrating for both the pet owner and the clinician. And it can also pose serious risks to humans as well, because oftentimes these parasites aren't just isolated to the animals, but can cross-infect into humans. We call these zoonotic risk. And this can cause some serious problems, especially with these reoccurring parasites, because as the parasite recurs in the animal, it can potentially cross-infect into the human owner or clinician as well. More than 200 parasitic species that we know of are zoonotic. And this can include some important ones like Cryptosporidium parvum, Echinococcus, Toxoplasma, and Giardia. And as these parasites spread, so do antiparasitic resistances. So, in the world of clinical parasitology, we treat these parasites with some common treatments. And overuse of these, especially of the antihelmintics, has led to increased drug resistance, which not only allows these parasites to recur more frequently, but also makes them more difficult to clear in the first place. Also frustratingly for global health monitoring is the global spread of parasites. So a lot of these zoonotic parasitic diseases blur host boundaries, and they can spread via vectors like ticks and mosquitoes. And their spread is being widely trafficked by human activities such as wildlife trade and animal relocation efforts. The veterinary impacts of this have been astounding. This includes emergence of parasites in previously non-endemic regions. So some of these parasites that used to be present only in certain locations are now spreading. So recently, more close to home, flatworm parasite of canines was recently found in Riverside County. And previously it had not been known to migrate this far west. And that also causes challenges for diagnosing parasitic infections. So if we don't expect these parasites to show up, they might not necessarily be targeted for in targeted diagnostic efforts. A classic case of this is leishmania, which the World Health Organization has recently shown has spread from its primarily endemic regions in East Africa and Southeast Asia to migrating north into Europe and being found more commonly in South America and Central America. And I want to take a step back and define kind of the diversity of eukaryote parasites. And taking us back to earlier biology, we define a eukaryote as any organism, non-bacterial, non-archaeal, that has a nucleus. It stores its chromosomal DNA within a nucleus. And this includes a broad range of organisms, including protista, um, oomycetes, and animals as well. So all these are considered eukaryotes. And parasitism is actually defined as a lifestyle, not necessarily taxonomic group. We can actually see that here because we see that parasites belong in the protista group, as well as the oomycetes. These are things like Pythium, our flatworms and roundworms. All of those have evolved parasitic lifestyles. And so the term parasitology and the lifestyle of parasitology has actually evolved several times throughout the world's evolutionary history. And parasitism is actually the most common mode of life. So if you separate modes of life in terms of parasitology, predation, saprobes, parasitology actually comes out on top as the most commonly employed mechanism by which organisms use to gain their resources. And there's thousands of species of parasite across diverse taxonomic groups. And they belong in the kingdoms Protista, Chromista, which includes our Pythium, Animalia, which includes our flatworms and roundworms. And so I'm really trying to give us a sense of the challenge here, which is how do you accurately identify all of these parasites to the species level using was really one of our biggest challenges here at MyDog, because we wanted to capture the parasites that belong to all these diverse organisms without relying on a limited PCR panel. And so to just showcase some of the limitations of PCR that we've noticed, PCR is widely used as a diagnostic tool to identify parasitic infections in animals, but really the problem is it has limited targets. So certain companies offer PCR panels with roughly 20 to 25 targets, and you can imagine how that might cause some issues, especially with lesser known parasites, right? If you're only having 20 to 25 targets, what are the chances that that is targeting a parasite that might be in your exotic animal species, in a zoo housed animal, for instance? And so that can cause false negatives, right? Because it ignores the more novel or less suspected parasites. If a parasite's not known to be endemic in a region, it may or may not be tested for due to a limited panel size. On the flip side, though, this can also cause false positives for many closely related species. If you have a species of Giardia that you suspect, you might get a false positive for that if your panel targets that Giardia species, when in fact it might be another closely related species. And so one of the challenges is how to simultaneously detect all parasites and pathogens regardless of the species. And you can already imagine, whoa, that would require a huge database of, ostensibly several thousand parasite species. How do we get that kind of database together? And this is actually more easily answerable than you might suspect, simply by asking the question, what do all parasites have in common? And the answer is actually very simple. All parasites have DNA. So all parasitic species have their DNA are composed essentially of DNA. In fact, only RNA viruses don't have genomic DNA. And so you can imagine if you're using DNA to identify parasites, you essentially can use a non-biased test to detect parasites simply because they have DNA, as all living things do. So this would actually cover your protista, your oomycetes, your flatworms like tapeworms, and your roundworms, but it would also capture bacteria and fungi as well. And so we actually call this an all-in-one test, simply because through looking at unbiased sequencing, we can detect all living things in a sample. Now, when we were first tackling this on about a couple years ago to develop this test, we started off with a number of research projects, and I'll highlight a couple of these, but essentially one of them involved the Cryptosporidium serpentis in eastern indigo snakes, which was a lesser studied Cryptosporidium species that causes often fatal gastrointestinal disease in snakes. We also looked at intestinal parasites of Hamadryas baboons, uh, a monogenea, which is a flatworm skin parasite of lemon sharks, cystoisospora, which is a protozoan parasite of black-footed ferrets, Tritrichomonas foetus in canines, and a larger collaboration with the University of Lincoln, Nebraska lab to improve genomic resources. And all of this was to expand our genetic library so we could be able to detect more parasites, add them to our database, and grow our database of parasites to essentially detect several thousand species. And I'm just going to highlight one study there, the Cryptosporidium and eastern indigo snakes. As I mentioned, Cryptosporidium serpentis is a deadly protozoan parasite of snakes. Prior to our work, it had no genome that was publicly available. And our collaborators at the Central Florida Zoo actually provided us gastric lavage samples of infected snakes. And through those samples, we were actually able to put together a genome of this parasite and screen for it in clinical samples. And one of the benefits here that I'm showing from using this type of unbiased metagenomic sequencing to sequence all DNA in the sample and characterize all of it is actually because we can study cross-kingdom effects. So we can actually show here with the same test, we can detect not only our parasite here, so we can see it, Cryptosporidium serpentis here showing up on the figure E here with the positive samples, but we're also showing other Cryptosporidium species, such as the chipmunk variety and Cryptosporidium hominis, which are usually they are zoonotic, but they're often spread by rodents. And so we can differentiate Cryptosporidium serpentis from those species. But we can also show in the same test bacteria, uh, viruses, and fungi using a single unbiased sequencing test. And this also helps us show putative associations between parasites and bacterial pathogens. Some parasites often work together with bacteria to worsen the symptoms of disease. And in our study, we identified Aeromonas hydrophila as one of the most abundant microbes associated with Cryptosporidium serpentis infection here. So we can see here that this microbe, Aeromonas, was primarily associated and mostly found in the presence of Cryptosporidium. And other studies have noticed that Aeromonas can worsen the effects of other Cryptosporidium diseases as well, such as Cryptosporidium murus in mice, can cause more severe necrotic damage when in the presence of Aeromonas. And so we do suspect that this sort of technology can shed light on these cross-kingdom effects and maybe illustrate that it's more than one player in these intense and very difficult protozoal infections. And recently, as a kind of exciting announcement, this paper was recently accepted as for publication at PLOS One. Now, through all those efforts, as I mentioned, we were slowly growing our MyDog database of parasite genomes over time. And just a couple months ago, back in March, we officially launched the all-in-one test, which includes parasites. And now through our efforts, we're actually able to detect thousands of parasitic species. But through this test, as we've done in our previous bacterial and fungal tests, even if we don't have that exact parasite in our genome database, we're still able to identify it to the genus level or higher if it's a novel species. So sometimes you might see in your tests Cryptosporidium sp, and I'll go through a few examples of this later. That means that's a novel species of Cryptosporidium that's not included or not known genetically, and hence probably a new species. We can launch this over all sample types. This would be fecal, skin, what have you. All those samples are testable through our new all-in-one test, and a broad host range. So we did focus on not just putting in parasite genomes from domestic animals like cats and dogs, but also things you might not normally have parasite tests for, like fish, avian, reptiles and amphibians, and dogs and cats as well, and marine mammals. And just to showcase how this test works, it really starts off with sample collection. So the clinician will collect a sample, be that fecal, skin, oral, what have you, um, and we extract the DNA from that sample, and all that DNA in that sample gets sequenced. We then compare um, that DNA in that sample against our internal library that we've curated of bacteria, fungi, and parasites. And that DNA is matched to our library of organisms. And that gives us a species level resolution. If that DNA from that sample exactly matches Cryptosporidium serpentis, it will be labeled as Cryptosporidium serpentis. And currently that database contains over 65,000 species of bacteria, over 10,000 fungi, 49,000 other eukaryotes. This would include things like algae. This would include things like parasites. Um, and 4,300 of those 49,000 genomes are parasites specifically. So that's a huge advancement over 25 targets. We are essentially giving over 4,000 targets here. And so again, as I mentioned, even if we don't have the exact parasite species in our database, if it's a novel species, if those reads are, or those pieces of DNA aren't an exact match to any of our species in our database, it will get classified to the next closest level. So maybe that'll be a genus classification or a class classification or family. So there will be very few, if no cases where we won't know anything about a parasite. We'll be able to at least tell you something about it, even if it's an unknown species that's not in our database. And just to showcase what this test has looked like since our parasite launch, um, we have received several samples since our launch, just simply back in March. And I'm showing here the pathogens versus the not pathogens and no eukaryotes detected. So here we're showing, we get several detections of parasites in our samples. Roughly, can be range from 10 to 20% of samples in any given sequencing run of over 80 samples do test positive for parasites. So just due to the fact of our broad species range of animal species we get receiving at MyDog, and our broad range of parasites that we are able to screen for, we can see some really cool novel things, and it reveals more detections than a limited panel of 20 to 25 targets might get you none detected. We can actually see that there are parasites in there that might have previously gone unnoticed. And just to showcase a brief schematic of what that kind of looks like here, here are our most abundant parasites. So not all of them, but our most abundant parasites we've seen divided up into our different animal types here. And you'll see some kind of cool ones. I like looking at this sort of data every now and then to get a sense of, oh, what new species are we seeing today? Again, we see a lot of Cryptosporidium SPs. These are unknown species of Cryptosporidium, which is always pretty exciting. We can see those in dogs, birds, cats, ferrets even, um, as well as some common things. So, Giardia duodenalis is a very common protozoal infection of canines and humans. And sure enough, we see that very often in canines as well. It's one of the most common parasites we see in dogs. Kind of surprisingly, the most common parasite we see is blastocystis, which is this, it can often be zoonotic, but it's this parasite of variable concern. Um, Sometimes it can exist in an animal and not cause any problems. Other times it can cause more severe GI distress. And then we can often see zoonotic species as well. I mentioned Cryptosporidium hominis, we are able to see that and frequently do see that in several animals. And so essentially what I'm showing here is we show species level classification of parasites, but we're not limited to a reduced panel, so we can show, several species here that may have been unnoticed, and this and their host range is incredibly broad too. So I'm just showing here even just a few animal species that we have received. We can get a very diverse set of parasites from those. And now I just want to take us through a few different case reports where we've seen parasites before, and kind of showcase some kind of interesting cases. So this was a case we received from a sheep early on in our parasite test. And, as our previous tests do, we still show the bacteria and the fungal pathogens, if any, are detected. But now, hey, we offer parasites. And so not only do we show that the parasite species, we also give their percent abundance. So maybe if a parasite is, only 50% of your eukaryote microbiome, but the other 50% is a healthy protist, maybe you'd wouldn't be as concerned about that parasite. But here what I'm showing is a Trichostrongylus, which is a zoonotic species of roundworm disease, often found in sheep. And in this case, it was 100% of the eukaryome. So it means 100% of the eukaryote profile in this sample belonged to this parasitic species of roundworm. And we were even able to give an estimated cell count for that at 150 cells. And with each of these samples and parasite species, we offer aid links. And that takes you to a helpful resource that myself and the other scientists here have curated to show a bit about, some interesting information about that parasite. So we'll offer information about, the taxonomic classification of this parasite, what its size and shape and color might be, as well as its zoonotic potential here. So we indicate here that this particular species of Trichostrongylus is in fact zoonotic and can transfect into animals. And in this case, here's kind of an example of what that parasite looks like. It's actually kind of a cool looking parasite with these little spots in the center. And so this can give us a great glimpse at what these parasites actually, additional context beyond just yes or no, this parasite was detected. Moving on to another case here, this was a pigeon. And again, we show that it had some rather concerning bacterial pathogens, including Corynebacterium diphtheriae. Um, However, a parasite was also detected within this sample, and that's the Ralliatina parasite. This is not a roundworm like our last case. This is actually a flatworm or a platyhelminth, and it's in fact a tapeworm. Um, and we can kind of see here what that sort of looks like. Again, we do provide aid links for this parasite. And this was in fact a novel species. So this was a species that wasn't in our database of 4,300 parasite species. However, because we have several Ralliatina species, we're still able to classify this to the genus level and get you the next best thing above a species level identification due to this being a novel species of parasite. One more case I want to show here is bringing this a bit more close to home, is a domestic cat. And this one actually tested positive for an oomycete parasites. Now we're taking this beyond our flatworms and roundworms and going with the same test, we're able to identify oomycetes like Pythium insidiosum, which is a really severe cause of skin disease, primarily in tropical climates or more swampy, humid climates. If anyone ever wants to, as anyone at My Dog can tell you, if anyone ever wants to talk to me for half an hour about what the difference between oomycetes and fungi are, I'm always happy to have that conversation. But they're technically not fungi. They belong to this group of oomycetes or eukaryote parasites. Here's an example of what that kind of looks like here. So it does look fungal in nature, but it is in fact not. And in this case, it's a very dense, hostly rich sample. This was taken from a lesion from the cat. And even so, with all that host DNA going around, we were still able to detect this parasite. And we determined that it was the um, only non-host sequence that was present in that sample, suggesting that this was in fact the cause of the lesions that this cat was experiencing. Now, earlier I showed all the different parasites that we've seen in our exotic and domestic animals, but even if we just subset what we see in dogs, you can already see we get a very broad range of parasites that we detect in canines alone. Um, Now, again, there's some more common species than others. So we see a very common one, again, is Giardia duodenalis. We see that very frequently in canine fecal samples. However, we also get some lesser common things like Cryptosporidium canis oftentimes shows up. A very common one we see is Trichomonas tenax, which we primarily get from oral cases where a canine is suffering from severe periodontal disease. This is an anaerobic parasite that lives underneath the plaque of canines with periodontal disease. We see that quite frequently as well as some more concerning zoonotic parasites like our Sarcocystis and Strongyloides as well oftentimes show up. So um, this gives us a broad sense of what we see even when we just focus on dogs. We have a broad range of parasites that are detectable through our test. Now, one really cool case I want to show that I think highlights uh, really the strengths of using a single test to identify bacteria, fungi, and parasites in with a single test in a sample is it allows us to look deeper than just parasite presence, yes or no. And a classic case of that is the canine heartworm. Um, Now, we actually recently received a sample that tested positive for the causative agent of heartworm, this parasite, Dirofilaria imidis. It was not at an extremely high abundance. We only saw 2 cells in that sample, but we know heartworm is not supposed to be there. They circulate their microfilaria or their small young worms through the bloodstream, and so they can oftentimes be difficult to detect. However, we still were able to detect this parasite species in this sample. Um, and but there's interesting stuff we know about this parasite, and one of those interesting facts is that it doesn't exist alone. Now, heartworm actually uses an endosymbiont bacteria. It houses a bacteria within its cells, and this bacteria allows the heartworm, to successfully reproduce and lay eggs. In fact, uh, this is the target for for the initial heartworm treatment. Um, uh, Essentially, clinicians treat the heartworm with doxycycline to first kill off this Wolbachia bacteria and slow or halt the worm's ability to reproduce and produce successful offspring, and then follow that treatment up with antiparasitic drugs. And so that's the first stage of essentially the treatment is killing off this endosymbiotic bacteria. But what's really fascinating is our test in that same sample uh, is able to detect not only the heartworm, but the Wolbachia as well. So we're able to show that we can see the entire picture in these complex multi-tiered infections. And so as a clinician, this could have a lot of value, right? You could submit a sample, tested positive for a heartworm, you see the Wolbachia, and then you start treatment, right, with doxycycline. Then maybe you submit another one after your doxycycline treatment, and then you see, does Wolbachia still show up? Um, and that could give a sense of, of how successful the doxycycline treatment was to halt the spread of heartworm. And so we do offer the ability to not only see the heartworm itself, but also the other bacteria that might be enabling this infection complex. So just in brief summary, the MyDog all-in-one test combines our bacterial, fungal, pathogen diagnosis with an equally expansive parasite test. We have thousands of targets here that we can screen for, um, and that library grows pretty frequently. So, it will continue to expand even beyond the 4,300 targets we have now. Since the official launch in March, we've detected over 90 unique parasite species and over 250 samples. So that means over 250 samples have come to us back parasite positive. And within those 250 alone, we've seen 90 unique parasite species, which is a far cry greater than the 20 to 25 targets that other diagnostic services typically provide. This includes common parasite species from domestic animals, but it also includes the lesser known parasites from exotic animals that maybe there's not a panel existing for that parasite yet. The all-in-one approach that we've launched provides a complete picture of parasites, fungi, and bacteria within a single sample using a single test. And I highlighted one unique case there, the Wolbachia heartworm detection, but there's several others that involve the cross-kingdom interaction. So maybe you'd want to treat the parasite, but you'd also want to get rid of the bacterial pathogens that are either co-colonizing due to the immune suppression of the host or maybe worsening the symptoms of the parasitic infection itself. So we do offer the ability to see the entire picture, and that can give a greater information that you as a clinician might need to treat this case. This is especially important for polymicrobial infections, right? So sometimes disease isn't one microbe, one cause. There can be several species that are co-enabling the infections in these cases. I mentioned Trichomonas tanax earlier, which is often we see that in the severe periodontal disease for canines, but oftentimes that's only one cause of a greater system at play, right? Those parasites need that anaerobic environment that's created by the bacterial pathogens that are causing the plaque buildup and oral disease in these cases. And so this can give a sense of the entire picture at play and allow a greater chance to successfully treat all the disease agents within a sample. And just to summarize here some of the benefits again of this all-in-one test, you get 4 tests in one, basically. You get all bacteria, both aerobic and anaerobic, all fungi, you get antibiotic resistances, parasite detection, within a 48-hour turnaround time. We do offer free shipment for these samples and free veterinary as well as microbiological consultation. And with that, I'm happy to take any questions.
Awesome. Thank you so much, Mark. That was great. Yeah, you're getting a couple claps here. We do have a few questions. The first one is, how do you distinguish between two parasite detection and environmental background or DNA contamination in these metagenomic samples?
That's a good question. I would say DNA contamination is not necessarily an issue, right? Because it's unlikely you'd see DNA from the parasite itself um, in a in a as part of an environmental contaminant. These parasites aren't often airborne like fungal spores can be. But to counter that, we do have detection thresholds that we provide, and we do provide bioinformatically several controls as well as wet lab several controls. So we're able to control for any contamination you might see. Now, environmentally acquired, those can exist, but they're unlikely to be colonizing. And so um, even if an egg is, essentially you scooped up feces from the ground, right, to put into the my dog tube, and maybe there was an egg or two of a parasite on the ground where that feces was collected, that's unlikely to show up in our report um, due to just the low abundance there. So it's also kind of a numbers game that comes into play.
Mhm. Beth is asking, will there be an option for only testing for parasites in the future? And yes, there will. We are planning to launch that probably in the next month or two. So that is coming very soon. Tiffany's asking, what sample was used for the Wolbachia and heartworm study?
Oh, it's a great question. So that was blood plasma.
Mhm. Could this approach eventually support the surveillance of emerging zoonotic parasites in wildlife or for environmental monitoring?
Definitely. That's a huge proponent of using this test because even if it's not a species that's commonly tested for, like you want to know what might be showing up here next year or the year after based off of early surveillance, that's where this test can really shine, is because we're not really condensed to a panel of targets that costs more to apply each individual parasite species. We have an entire range there of parasites, and so we essentially test for everything in a sample, even if it's, let's say, something that's not known to be endemic in that region. We still test for it, and that can be powerful because, as I mentioned, these parasites are spreading, um, and uh, this gives you the chance to see those, even if clinically they might not be widely tested for yet in your region.
Hmm. I guess a more personal question. What was the most surprising parasite you've detected so far? Was there anyone that stood out to you?
Ooh, yeah, actually, there have been a couple. I would say that we recently received a sample that had monogenea, which is a flatworm, in an eel, which is pretty cool. Um, and it was an unknown species of monogenea, which is kind of always exciting for me to see new species. Another one was um, it's a aquatic oomycete called Aphanomyces. And interestingly, we've been seeing that in turtles when it's primarily known to be infect fish. And we've been seeing that in axolotls as well. Um, Interestingly, axolotls and turtles suffering from skin lesions, which is the exact symptom or sign it causes in fish. So we might be showing that the host range for some of these parasites is broader than previously respected.
Hmm. And you've been mentioning that sometimes you can't assign a species level. So these taxonomic assignments can be challenging because we have such limited genomic reference databases. But what kind of cutoffs are being used when you can't say, oh, it's this species or this genus?
Yeah, so that's basically a genetic identity cutoff. Now, the exact numbers are variable between each species, but um, for genus level, it would be a non-specific hit that's equally similar to maybe 3 or 4 different species. We might back that off to the genus level, especially if it's less than, let's say, 90% similar or 95% similar, right? Bear in mind, like humans and chimps are around 90 to 95% similar. And so you don't want to overcall to the species level when you can't be confident that it is such. So we do heavy vetting of those. So we're not incorrectly giving you the species identity based off of percent identity of the DNA to its closest hits there.
Mhm. And last question, how does the sensitivity of the MyDog test compare to more targeted PCR for known parasites like Giardia or Cryptosporidium?
That's a good question. We have ran some tests where we've compared our results to the PCR tests from clinical samples, where the PCR has said, yes, parasite. We ran that through our tests as well. We had a pretty substantial sample size of those. I believe we had over 50 positive cases to compare with. Every single time that those tests reported a positive, we were also reporting a positive. Most of the time they reported a negative. We would also report a negative, except in cases when uh, those samples reported negative and we see a parasite species that was not part of their target panel.
Mhm. Yeah, if I remember correctly, we actually published one of those case reports. It should be available on our website. Yeah, and that's all the questions we had. Thank you so much, Mark. You did fantastic. Thank you for everyone who joined us. If you would like to rewatch this video or share with anyone else, it will be on our website shortly. So thank you and have a great rest of your day. Goodbye.